In this module, I want to cover the cell life cycle and the difference between mitosis and meiosis. The cell life cycle is divided into two broad periods, interphase and mitosis. Interphase includes G1, which stands for gap 1. Gap 1 can be the most variable in duration of all the phases. In quickly growing cells, it can last for hours, and in slowly growing cells, it may last for days. It seems that when the cell gets large enough, where its surface area to volume ratio is compromised, that this is the trigger for the S phase, which stands for synthetic phase. During S phase, the DNA is replicated, so the DNA content is doubled, but the number of chromosomes remains the same. During G2, the cell makes the final preparations for mitosis. Organelles have been replicated so that each daughter cell will have enough of each organelle. The centrioles finish replicating, and enzymes that are necessary for mitosis are made. Mitosis includes phases for splitting the nuclear contents, the chromosomes, followed by a phase for splitting the cytoplasm, cytokinesis. Each phase of mitosis will be addressed through the remainder of this lecture. It is important to understand that checkpoints occur in G1 and G2 to ensure that the cell is ready to divide. In your gene expression module, we will discuss how the rate of cell growth and division are regulated by proteins called tumor suppressor proteins and oncogene proteins, and how these proteins can fail, and then cancer ensues. Which cells in our body divide by mitosis and why? Many cells are mitotically active. The squamous cells in your epidermis, hair cells, cells lining your GI tract, etc. Cells are mitotically active in areas that experience a lot of friction or damage. We need cell turnover to replace aged cells or areas that have been damaged, say from a laceration, or cells that have been damaged, say cells lining the lumen of the stomach and have been exposed to the acid. It is also important for cells to divide if they get too big. If they enlarge too much, their surface area to volume ratio decreases, and this decreases the rate of diffusion for oxygen, carbon dioxide, and other metabolic molecules. The website I have posted here has a lovely tutorial for mitosis and meiosis, and that is where I got the picture, too. I want to reiterate that before the cell can divide, it must replicate its DNA. Remember, DNA is the code for protein synthesis. Each daughter cell will need its own genome to drive gene expression and have the necessary arsenal of proteins in order to function. DNA is replicated in a semi-conservative fashion, which means that the chromosome unwinds and each strand serves as a template for a new strand. At the end of S phase, each chromosome has doubled, each one consisting of 2 sister chromatids. So there are still 46 total chromosomes in the cell at this time, just each one has 2 sister chromatids. At the end of interphases G2, the mitotic phase begins with prophase. During this time, the important processes to remember are the chromatin starts to condense and the nucleoli disassemble. Late prophase is really called prometaphase, and during this time the nuclear envelope disappears and the mitotic spindles, made of microtubules, start to extend from the centrosome and attach to the centromere of each chromosome. The centromere is where the sister chromatids are attached. In metaphase, think M for metaphase and M for middle because the chromosomes line up in the middle of the cell along the metaphase plate. For anaphase, think A for anaphase and A for apart. It is during this phase that the centromeres are split and the chromosomes are moved to the poles of the cell, and they look like V-shaped as they are pulled from their centromeres. The last phase of mitosis is telophase, and in this stage the nuclear membrane returns, the nucleoli return, the mitotic spindle disassembles, and the chromosomes uncoil back to the chromatin state. Cytokinesis involves the synthesis of a cleavage furrow where contractile proteins squeezes the cells apart. At the end of mitosis, there are two diploid daughter cells, each with 46 chromosomes. Each daughter cell is identical to each other. In the next two slides, I will discuss meiosis. As I talk about the process, I want you to note the following. Meiosis only occurs in the gonads, ovaries of women, and testes of men. It's different from mitosis in that there are two phases of meiosis, 2 times when the nucleus is split. There is a crossing over of homologous chromosomes during prophase 1 that gives great variability to the daughter cells. No two will be identical, and the end result are 4 daughter cells that are each haploid, meaning they have only one copy of each chromosome. So each daughter cell has 23 chromosomes. During prophase 1, synapsis occurs between the 2 homologous chromosomes, and they form a tetrad. This is where crossing over occurs, which means that pieces of chromosomes are being swapped out between the homologous chromosomes. This phase lasts the longest of all the meiotic phases. As in mitosis, the chromosomes condense and the nuclear membrane and nucleoli disappear. Metaphase 1 is different from metaphase of mitosis in that the homologous chromosomes pair up in the middle instead of end to end. Anaphase 1 involves separating the homologous chromosomes, but the chromosomes are not broken at the centromeres, so the sister chromatids still remain attached. In telophase 1, the chromosomes unwind into chromatin, and cytokinesis is called interkinesis. The 2 daughter cells are called haploid in that they have only one copy for each homologous chromosome, but there are still 2 sister chromatids. Therefore, a better term to use is pseudohaploid. Meiosis 2 begins with prophase 2, in which the nuclear membrane breaks down and the chromosomes condense again. In metaphase 2, the chromosomes align at the middle of the metaphase plate. During anaphase 2, the sister chromatids are separated. Finally, at the end of telophase 2 and cytokinesis, there are 4 haploid daughter cells, and they are each genetically diverse from the stem cell, or mother cell. I encourage you to watch a few videos so you can see the difference between mitosis and meiosis. This first website has a flash player to compare mitosis versus meiosis, as well as a downloadable file for Mac or Windows. This second site has many good movies. Click on either mitosis or meiosis to watch them step by step. There are many other good websites that I have included in a worksheet I posted on mitosis and meiosis. You do not have to complete this worksheet and turn it in. It is merely posted as a review.